briefly introduce Professor McCarthy. Let me share you his CV. So Professor McCarthy received a bachelor degree in pharmacy from Ohio Northwest University. Northern University, and PhD in medicinal chemistry from University of Georgia. So he's currently working as a professor in the departments of medicinal chemistry and pharmaceutics, the Frank A. Duckworth Eminent Scholar Chair, and also as a director of UVCT's SI Translational Drug Development Corps, University of Florida College of Pharmacy. So he's trained pharmaceutical scientists and pharmacists whose research focuses on the design, synthesis, and the development of drugs to treat pain, anxiety, and substance abuse. And he's also an internationally recognized expert on Kratom. So without further ado, maybe we can start to hear the lecture from Professor Chris. So, Professor Chris, the time is yours now.
Thank you. Can you hear me okay?
Yeah, I can hear clearly.
Perfect. Okay, let me put on the slideshow and we can move on. All right. So thank you very much for inviting me to come and speak with you. I'm sorry I can't be there in person. It would be my preference to come and enjoy some of the best food on the earth, I think, in Indonesia, but I'm stuck in Florida. So
we can invite you to come to Indonesia.
I will be coming in next year. There's a conference in Thailand, so I'm hoping that I will visit Thailand, Malaysia, and Indonesia on the same trip. So
okay, perfect.
Hopefully we'll be there.
Yes, thank you.
So I want to start not only by thanking you all for visit with you today, but I want to thank everyone that's involved in the data that I'm going to present today. So I'm very fortunate that I get to lead this group of very talented scientists, and they do all the work. I get to write the grants, write the papers, and present the work that they get to do. But I really want to thank them before anything else. So I have a very great team of chemists that have worked with us for quite some time now, including one that's from University of Malaysia, Nelson Cheer, who's listed there under the chemist group. He's visited with us a couple of times. He's still working on his PhD in Malaysia, and he should be finishing up in the next, actually in the next 2 months. And then we hope to bring him back to the US to work some more. But we've had a great collaboration with the University of Science Malaysia and getting some science behind not only the work that they've done there, but being able to share that science internationally. And that's a goal, hopefully, that we can all share together and work together to bring some science from Indonesia to this party. We've done a lot of work on receptor and functional studies. We've done a tremendous amount of work in drug metabolism and pharmacokinetics. Most of that has been led by Bonnie Avery and Abhishek Sharma. And then several students finished and graduated and moved on. But we also have a huge team of behavioral pharmacologists. Lance McMahon and Jenny Wilkerson are really leading that team. They just recently moved to Texas Tech University, and we miss them being down the hall, but they had bigger and better opportunities. The last thing that I think is one of the most interesting pieces that we have at the University of Florida is in the United States, the university is structured in such a way that usually you have health sciences at one university, and then you have the agricultural school at another state-sponsored university. In Florida, this is a very unique situation where we have both the science center as well as the agricultural school within this campus print, and we have a tremendous collaboration with our horticultural scientist, Brian Pearson, who leads that effort with Swasi Nadakudi, also working from plant genetics side. And so I'll mention a little bit about some of the work we're doing in understanding the cultivation requirements for Kratom as well. We've been extremely fortunate to have a tremendous amount of funding from the National Institutes on Drug Abuse as part of which is one of the institutes under the National Institutes of Health. And we've had a lot of sponsored funding from various marketers of Kratom in the United States. So it's a very sources of funding that we've had, from government to private industry, and also donations to the university from many individuals throughout the United States. And we're also fortunate we have almost 60,000 students at the University of Florida, and many of those are looking for volunteer opportunities. And so we've had a lot of free student volunteer work, which has really helped this forward. So I'll start just to put us all on the same page, which we already know tremendous amount about, and that's Mitragyna speciosa, of course, is in the Rubiaceae family, and it's a tropical tree found native, particularly in Malaysia, and now widespread throughout Indonesia as well. As we know, most of our supply in the U.S. is coming from Indonesia. Referred to as Kratom or Kratom, and biak or Kitom in Malaysia. There's about 40 different alkaloids that have been isolated to date. Some reports push that number up towards 50. In all honesty, have a hard time knowing if it's really 40, 50, or 20. And that's mainly because many artifacts during the isolation process. Chemistry can happen during all the harsh conditions of acid-base extraction and chromatography, but we certainly know that there's a abundance of alkaloids, primarily 15 to 20 that we can isolate on a consistent basis and look at. And you all probably know this, but I found this a very fascinating fact that the whole genus of Mitragyna was named based on the shape of the leaf, and the leaf shape being that looks like a bishop's hat or a mitres hat. And so if you take the leaf that's at the top of the page there and sort of rotate it counterclockwise 90 degrees, and put a little head under where the stem is, it kind of looks like a hat. And it looks like a hat or a mitres hat, and that's where the name Mitragyna came for the genus. And all of the plants in the genus Mitragyna have that same shaped leaf. One of the differences in the species, though, is aside from chemical content and chemical fingerprints, is actually the flower. And so the flower of Mitragyna speciosa is a nice yellow color, very fragrant, very attractive smell, and we have not really pursued much of the phytochemistry that's in the flower pods, which then turn into the seed pods. But there is some reports in the literature on concentrations of alkaloids even within that flower pod. So won't tell you too much about the kratom use in Southeast Asia, as you are more familiar with this, although it does vary from Malaysia, Thailand, and Indonesia. I'm going to speak about it more or less around the way that it's used in Malaysia. This is where I've been on the ground, and I've actually visited these places. These are photographs that I have taken in this roadside stand that you see down at the bottom of the center of the screen there in Malaysia. Interestingly enough, this is an illegal product to sell in Malaysia, but you can stop on the side of the road and purchase it pretty much anywhere out in the country that you can find it. Also, there's a tree essentially in every yard of every home that I've seen in the border region of Malaysia and Thailand. And of course, this has been used for centuries for pain relief, as well as stimulant to improve work capacity, and in some instances to reduce opioid withdrawal, or as a way to keep individuals that were on opium from going into withdrawal as they would wait for more opium to come to them. Recently, polydrug users in Malaysia, those that are using opioids as well as methamphetamine, have started to use Kratom to reduce their use of methamphetamine as well as opioids. And so we're starting to look at this. One of the major problems in the United States, aside from the opioid crisis, is the fact that we have absolutely no pharmacological treatments for methamphetamine, cocaine, or any other psychostimulant abuse or addiction or misuse problems. And so this is a major interest with the National Institute on Drug Abuse here in the United States. And so what you can see here down on the left, of course, is a tree in the flowering stage. We were lucky enough to be there and capture that at the right time of year. And then in the top right-hand corner, the traditional way that it's served in Malaysia is leaves are plucked fresh off the tree, ripped in pieces, and then boiled in water for 2 to 3 hours, and then served in to-go bags or plastic bottles, and they're taken off for the rest of the day. And that's a 1 day's supply, essentially, that will be divided up into 3 doses throughout the day for morning, afternoon, evening. The predominant ingredient in Kratom, we have Mitragyna. This is a quinantine-based indole alkaloid, and it's a very interesting model. When we look at it, one expect this to have opioid-like activity, just from a structure perspective and from a medicinal chemistry standpoint. And so this was one of the things that was really fascinating and interesting to us, was how is this molecule interacting with opioid receptors, and what can we learn from this in time. And what we've learned is it doesn't just interact with opioid receptors. I think that's been one of the more interesting pieces of the puzzle in unraveling this. I will say that there's a huge difference between Kratom use in the United States and what we see in the traditional setting. So in Thailand, I know that the leaves are primarily chewed, and in Malaysia, as I said, more commonly is brewed into a tea. In the United States, it's a very, very different product. It's not fresh. It's leaves that have been dried and ground into a powder. And those leaves have gone through senescence phase. And so they've chemically changed from what they were when they were picked fresh off the tree. These have dried. They've been exposed to air, humidity, and other factors like UV light after they've been separated from the tree. And all of those are chemical environments that will change the chemistry within the plant. And what we've seen in products that are in the United States and available in the United States are many of these oxidized alkaloids that we can isolate from this dried plant material. It's very available in the United States, particularly in what we call smoke shops or head shops or vaping shops. This is not a typical store like you would imagine a pharmacy or a health food store. These are being sold primarily in locations where we can find tobacco products or CBD cannabis type products. Alongside of that is Kratom, and so it's carried a sort of black mark, if you will, or a dark side to it in the United States from the beginning, because this is where it has been sold. It hasn't been sold along your traditional herbal products. The American Kratom Association, AKA, which has been very involved in Indonesia, reported a few years ago now that there was about 2,000 metric tons being exported to the United States every month. And if we look at what that means, it essentially says that there's more than 15 million users in the United States. And you can imagine what a market that is for products, particularly that would be attractive to farmers and growers in Southeast Asia. What you see there in that picture below is what the product looks like, very different from what we saw on the previous slide, where these are formulated into capsules or tablets or concentrated extracts and energy shots. In some cases, there's a small product back here called MIT 45, which is a highly concentrated oil-based extract that has 45% mitragyna within it. And so these are very concentrated, very potent compared to what we would normally see in traditional use. Also, products in the United States are marketed really more towards a botanical bend. So you have some products that really look like they are botanical, and they'll say red vein, Mitragyna speciosa, or premium Bali blend, or something that sounds much more like an herbal product. But at the same time, we have marketing for products that are packaged in capsules and say Foria. So kind of giving the buyer that, or the shopper, the idea that is going to cause a euphoric event or a pleasurable experience. We have some products called Viva Zen, which make you feel like life is great. And so there's some confusion in the marketplace, and this has led to a lot of the issues that the FDA, the administration, and our government has really created around this product, and wondering if it's truly safe for everyone to use. And so that brings the question of, is Kratom really a threat, or is it a therapeutic? Anecdotally, we know it's been used even in the United States for chronic pain, mood elevation, and for opioid use disorder. We're in a tremendous crisis in the United States where almost one person per 5 minutes is dying from opioid overdose. You see the numbers, or maybe you haven't seen the numbers, but for the first time ever last year, the United States eclipsed 100,000 deaths in the year from opioid overdose. And if you look at the numbers, it really comes down to about one person every 5 minutes. We've also seen that Kratom products have been adulterated or contaminated with pharmaceuticals or with other illicit substances that can make the product much more potent, much more dangerous. In some cases, these are actually prescription drugs like morphine or hydrocodone. We've also seen adulterated substances with fentanyl derivatives, and so this is frightening from the standpoint that if people think they're buying a product, it could be laced with other potent, more deadly substances. There have been a lot of claims in the United States that Kratom is severely addictive and deadly. In fact, just last week, there was a report from Atlanta, Georgia, a family that their son overdosed, supposedly only was taking a Kratom product, overdosed and died on that product. And so it's still in the news in the United States. There's still a lot of concern over the danger and the harm that these products can cause. And again, I bring you back to what I just mentioned. If you look at what products are available in the United States marketplace, many of them are these highly concentrated extracts that people are ingesting, which could be 40 to 50 to 100 times more concentrated than what a normal dose of Kratom would be in a traditional setting. As you're probably aware, the World Health Organization also took steps to consider a ban internationally at Kratom. This was rejected near the end of last year. I was actually part of that panel that presented data to the World Health Organization, the United Nations, to show that there was a lot of strong science coming around, not just from our labs, but from labs across the globe, and really putting some science behind the fact that all these anecdotal claims that have been made for centuries have some legitimate science behind them now. You can see this article here from the USA Today, which is a huge newspaper here. Herbal drug Kratom linked to almost 100 overdose deaths from our Center for Disease Control. It's interesting if you would actually read this article. At the very end, it says the majority of those cases, the Kratom was found, the blood samples for those deceased individuals also contained nil. So, the media has sensationalized this a little bit, also trying to get regulation around these products. And Kratom is illegal in 6 states still to this day: Alabama, Arkansas, Indiana, Rhode Island, Vermont, and Wisconsin. It is completely outlawed in those states, sales and possession. And it is outlawed in certain towns and counties in various other states throughout the United States. So there are some restrictions and control around it in many parts of our country. So what is the big deal? And we think that, based on case reports, very few deaths are attributable to Kratom alone. We know Mitragyna, that major compound, the major alkaloid within the plant, is a partial mu-opioid agonist. So it is interacting with the same targets that morphine or heroin interact with, but it's doing it at a much lesser activity. So it only creates about a 40% maximal response. And so if we look at activation of G proteins, and 100% would be the maximum. Here we have Damgo, which is a peptide, an opioid peptide that produces 100% response. We can see Mitragyna actually can only produce about 40% response in this assay. And so I kind of equate this as to a rocket ship versus a bicycle. You have something that can get you all the way to a maximal effect very quickly, something like Damgo, which would be comparable to heroin or morphine, and then something like Mitragyna, which gives you some activation and moves you along, but it doesn't really give you that full activity. The other thing that has been interesting from a standpoint of cellular biology and activation is that we consider Mitragyna to be a biased agonist. In other words, it does not recruit a protein called beta-arrestin, and beta-arrestin is associated with many of the negative side effects linked to opioids. And so molecules that are biased agonists that don't recruit beta-arrestin are under investigation and development as opioids with low respiratory depression and low constipative effects. It doesn't mean that their abuse potential isn't all similar. All opioids will have some abuse potential attributed to them. But what we're seeing here is lower respiratory depression, lower constipation effects, and potentially lower tolerance development effects, all which would keep doses low enough that you wouldn't press into some of these side effects that are dose limiting on the clinical side. There are a couple of molecules, one TRV-130, olaceridine, which has been approved by the FDA now in the United States as an opioid for pain treatment. It is a biased agonist that performs in this manner. But interestingly enough, I was also on the FDA panel that considered that the safety profile was actually no different for this molecule than it was for other opioids. And so at the first time it went up for approval, it was denied approval. Eventually it approval on a limited basis for certain situations. There's another molecule that's in development called PZM21, and it's thought to be an even more selective biased agonist. And so we will see what the results are. But the bottom line is that we think mitragyna follows this pattern, and mitragyna by itself, once isolated from kratom, could be an interesting molecule for treatment of pain or potentially treatment of use disorders. And so if we look at it from a therapeutic perspective, we think kratom, and not just mitragyna, but kratom as a whole, has a potential several medications that are used during opioid detoxification. Many of these are targeting not only opioid receptors, but adrenergic receptors, such as alpha-2 receptors, where we see drugs like clonidine or lofexidine that are being prescribed as treatments for opioid detoxification. We also see other analgesics and anxiolytic drugs that are employed for this opioid detoxification. And we think that there's great potential for kratom to be used as a single substance, although it's a very complex botanical mixture of molecules. But if this could be utilized as a tea or even as a capsule or some sort of medication, that adherence and chances of completing detoxification would be much easier for these patients. They had to take one thing rather than a handful of medications at various times. We also look at this as a potential medication-assisted therapy, or what we call as MAT in the United States. And this would be somewhat comparable to drugs like methadone or buprenorphine. And kratom is informally used to reduce opioid use. We've seen that through several surveys. We've seen that historically through traditional use in Southeast Asia as well. What we do know is that withdrawal from kratom, at least in the majority of cases, is very mild. And I show here less than 9 on the SALS scale. And if you're not familiar with the SALS scale, it's a subjective opiate withdrawal scale where people or patients individually report their withdrawal symptoms. And this scale goes from 0 to 30. And anything less than 10 or less than 9 is considered to be mild. Caffeine would be in this same category. So coffee or tea from traditional sources would be in this mild withdrawal category where kratom is. From sort of 11 to 20 is moderate withdrawal, and then from 21 to 30 is considered severe withdrawal. And this obviously looks to be very benign in terms of the subjective opiate withdrawal scale. And we do see polydrug users, as I mentioned earlier, using kratom to reduce methamphetamine. Now, the problem in the United States has been that we have very rigorous clinical trials in order to support therapeutic claims that have to be performed. And one of the major setbacks and drawbacks has been a lack of a standardized product that's available in the United States to conduct these rigorous clinical trials. We don't have the ability to really where materials are coming from. We don't understand what the chain of custody has been. Most of the products in the show up at some port, and they are then sold to various vendors, and then those vendors repackage the material and put them into their own branded products. And so it's very hard to know where products have come from or how they've even entered into the United States. So having a clear importation path would be helpful, but as mentioned, the Food and Drug Administration has banned importation into the United States of Kratom products. Yet that doesn't stop it, because I also mentioned earlier that we have 2,000 tons coming into the United States every month. It's probably much more than that. That was a pre-COVID number. And so, oops, I jumped too fast. So I mentioned that we have the horticultural school, the agricultural school affiliated with the University of Florida. Our climate is not quite the same as it is in Southeast Asia. We are humid. We do get a lot of light, but our soils are not nearly as rich, not nearly as moist. We basically have sand and clay-based soils, which make it difficult to really compare growth conditions in Florida versus Southeast Asia. But what we can do is we can grow these trees. All of these trees, interestingly enough, we don't know where they came from. They have been donated to the University of Florida by various users within the state of Florida, who somehow acquired a tree either through the internet or where, we don't know. And those trees, we've spent a tremendous amount of time doing DNA barcoding on and authenticating back to standards that we've been able to receive from Thailand or Malaysia. Would love to have some Indonesian products to see how very closely related the trees that we have are to what's growing in Indonesia. Interestingly enough, we found that there's basically at least two chemotypes of trees, one that's a high producing Mitragyna strain and one that's a very low producing Mitragyna strain, something that we almost equate to what we see in the cannabis field, where you have high producing THC cannabis sativa and almost zero THC producing cannabis sativa that we refer to as hemp or industrial hemp, which is really a fiber-based crop here. And so we're seeing this huge diversity in alkaloidal expression within Kratom. And we wanted to explore that further. When we were in Malaysia, we actually harvested from single trees within a farm, and we found that diversity of genetics even within those single farms. So we had some trees that were producing high levels of Mitragyna and some trees that were producing almost no Mitragyna within them. So this is a fascinating piece of information, and we have some theories as to how this may have happened or what's going on in terms of some genetic diversity and crossing, and how these kind of link even towards Mitragyna diversifolia, another species in the family. So we do know that there's a tremendous amount of alkaloids, as I mentioned at the outset, and these are 12 alkaloids that we have been able to isolate in fairly large quantities, and that's the 12 that are sort of blocked off on the left side. There's 3 compounds on the right side of the screen. I'll talk about that in a moment. The majority of reports suggest that Mitragyna is the major alkaloid. It's interesting, if you go back and really critically look at this literature from the 60s and the 50s, even before that, you can find that the original study by Shellard was done by sampling of 3 trees. And that 3-tree sampling indicated Mitragyna being at 66% of the alkaloid content. At that point in time, they also quantified, and my guess is they quantified Mitragyna, speciogynin, speciosialatin, and mitracylatin all lumped together. So mitragynin actually has 3 other isomeric pairs, where we can see here speciogynin. Maybe I should get my pointer, I'm sorry. So what we can see here is speciogynin, which just differs in the stereochemistry of this, this 20 position. And then we can see speciosialatin, which differs from mitragynin in only the 3 hydrogen positions. So this is in a beta configuration, whereas in mitragynin, it's in the alpha configuration. If you look at a liquid chromatography mass spec method, all of these compounds will more than likely coalute, and they could be over quantified as being mitragynin or the single compound. So it's taken time to really dig in, separate out all these compounds, establish methods that can really define all these different isomers that are present. And I think what's really fascinating is you have these 2 classes of chemicals. So you have these indole-based chlorinampheidine alkaloids, and then you have these oxindole type alkaloids, which are at the bottom here. These would be, metabolites of the compounds above them. So these undergo semi-pinnacle rearrangements to then form these isindole, oxindole type products from the indole products. So there's a lot of chemistry that's happening in the plant, and it's very fascinating. If we look to the far right column, one more thing I want to touch on before we jump off chemistry, and sorry, I'm a chemist, but I have to point this out. All of these compounds, just about all of them, possess this, what we call beta-methoxyacrylate. So this portion of the molecule at the bottom, they all are basically present in each of these molecules. Ring, but for the most part, you see this questions as to why would the plant consistently keep this? It must be some type of warhead or some type of interest in the plant to produce that. It actually could serve as an antifungal type role. These are similar to a class of natural products called strobilurins, and they have antifungal activity with a beta-methoxyacrylate moiety. We have studied these on human fungal pathogens, and we haven't seen any activity there. We have looked at some plant pathogens, and we've seen some mild activity there, but it would make sense that these could be antifungal and excreted from the trees, because the trees are growing in very swampy, humid, moist conditions. And in order to survive in those conditions, it would be favorable if they had something to sort of protect them. And so we think that might be what the actual role of these alkaloids is from an environmental standpoint. I'll jump over to these 3 compounds on the far right. We hear a lot about 7-hydroxymitragynin naturally occurring in the plant material, but from all the samples that we've taken, whether it's been from Thailand or Malaysia or even from the trees that we've grown in Florida, there's actually some farms that have started to develop in Hawaii as well, and we've gotten some samples from those farms in Hawaii. We don't see 7-hydroxymitragynin present in fresh leaf material. It seems to be a post-harvest oxidative artifact that we see, and we do know that it is a metabolite of mitragynin in animal species as well, including humans. And so the liver will convert mitragynin, also the intestine will convert mitragynin into 7-hydroxymitragynin. And why is this important? It's important because 7-hydroxymitragynin is a fairly potent opioid itself, and I'll talk about that in a few more minutes. We've also synthesized many of the other 7-hydroxy compounds that relate back to some of the major indole alkaloids within the plant to really study what these metabolites might be contributing pharmacologically. And then last but not least, Mitragyna pseudoindoxyl, which has also been apparent in the literature. This was originally reported as a fungal metabolite. So Mitragyna was fed to fungus or yeast cells and converted into this product Mitragyna pseudoindoxyl. It's not a naturally occurring product from the plant, and we've never seen it in the plant material itself. However, it's interestingly a metabolite that is produced in animals, including humans. And what we know is that it's produced from this 7-hydroxy metabolite. And so we see mitragynine converted to the 7-hydroxy mitragynine in both the intestine and in the liver. And then in the human plasma, as well as other species plasma, we see conversion from the 7-hydroxy mitragynine into this mitragyna pseudoindoxyl. So this becomes important as well because it's even more potent as a mu-opioid agonist than the 7-hydroxy mitragynine. So all of these were concerns for carrying a Kratom product forward and understanding what the ramifications of these metabolites are, and I'll touch on that here in a moment. We also embarked on a pretty aggressive screening of all of these alkaloids that we isolated through 82 different central nervous system drug targets. And what I'm showing you here is mitragynine through this 82 panel screen, where we did it at 10 micromolar, and anything that displaced the radioligand from those targets greater than 50% was considered an initial hit. And those were followed up on screening at 100 nanomolar. And what we looked for there was, again, a displacement of 50% of radioligand or greater. And what you can see here with mitragynine is it engaged a lot of targets at 10 micromolar, including serotonin, adrenergic, dopaminergic, as well as opioid channels, or receptors. But once we moved that dose down, or that concentration down to 100 nanomolar, we didn't displace radioligand at 50% at any target. And so in reality, this molecule seems to be pretty benign and not very potent at any particular receptor. We did want to interrogate it a little bit further, and we conduct binding affinity screens in our labs to see exactly what the affinity constants were at opioid and adrenergic receptors. And we do see that at mu and kappa opioid receptors, we have 150 nanomolar activity. We also see at alpha adrenergic receptors that we have micromolar affinity from 1 to almost a little more than 9,000 micromolar. And so is that really relevant from a standpoint of taking a substance? And yes, those concentrations can be achieved in vivo with traditional dosing. And so we do believe that there is a component of alpha adrenergic activity coupled with the opioid activity. We do see partial agonist effects at those alpha adrenergic and opioid receptors. And interesting, in the United States, poison control centers have been reporting overdoses or calls that have been coming into those poison control centers where people have ingested too much Kratom, and they are saying that much more resemblant of a stimulant overdose and not opioid overdose. These individuals are very excited, very aggressive. They're very in a sort of anxious state, a panic. They in some cases have gone into convulsions or high blood pressure states. And this is very different from anything we would think of with an opioid, where you would consider respiratory depression or sedation to be the main drivers of an overdose from those settings. And so we think that in very high doses, the stimulant actions take over, which is different from what we hear in the use of these compounds, or the use of kratom, because kratom usually is used at low doses for its stimulant properties and at higher doses for its more sedative properties. And so this goes a little bit against what we're at least in the United States as well. I do want to touch a little bit more on mitragyna, since it is the major alkaloid. And what we've done is looked at this in terms of its analgesic assessment. And we have seen in mice that it be a very potent analgesic at a fairly high dose. And so what you see here is percent anti-nociception on the y-axis, and then time across the bottom here. And at the highest dose of 100 milligram per kilogram in wild type mice, we get very potent anti-nociception through subcutaneous delivery. That's not very long lasting. After about 30 or 40 minutes, it's down to about 50% efficacy, and then it sort of tapers off after that. We do know that it's pretty much associated with the mu-opioid receptor, though, because we take knockout mice with the mu-opioid receptor, and at the same dose, we see absolutely no analgesic activity. Most pharmacologists would say, well, that's the end of the story. We know it's completely opioid-mediated analgesia. Our collaborator and I had a good long discussion, and we decided, let's push the dose up even higher and higher and see what we can find out. And sure enough, once we got to 200 milligram per kilogram, so we doubled the dose, we actually started to see almost 40% anti-nociception coming back in there. And what we were able to attribute that to was actually alpha-adrenergic analgesias. And so we dosed those same animals that were getting 200 milligram per kilogram, those knockout mu-opioid receptor mice, with yohimbine, an alpha-adrenergic antagonist in pink. And you can see that the alpha-adrenergic antagonist reversed that analgesia that we saw. And so we think that the analgesic activity and maybe all the pharmacological activity of mitragyna is associated to opioid and adrenergic receptors, both. One of the most complete studies that's in the literature, even to this day, 50 years later, was a study by Mako et al. At SmithKline in French. They had isolated mitragyna and taken it into full development at SmithKline and French Pharmaceuticals, looking at it as a comparator to codeine. It had great anti-nociceptive and antitussive activity similar to that of codeine, but had less side effects than codeine. However, they abandoned development of mitragyna as a pharmaceutical because at the same time, the non-steroidal anti-inflammatory class of drugs was starting to emerge, and it had no opioid activity and no side effects, at least at the time then. And so they decided not to pursue something that was still having opioid-like effects. So I mentioned 7-hydroxymitragyna, and this, in all honesty, is one of the most selective opioid compounds I've ever seen in my life, and I've been working in opioid chemistry for almost 30 years now. I've never seen a compound screened through that 82 screen at 10 micromolar that only touches mu, kappa, and delta opioid receptors. It has no other affinity at any other targets, even at 10 micromolar. And so we interrogated this as well to get affinity values at mu, kappa, and delta opioid receptors, and then functional activities. And we see that 7-hydroxymitragyna has pretty strong effects at the mu opioid receptor as an agonist. We wanted to also see if mitragyna itself and 7-hydroxymitragyna is substituted for morphine. And so this was a study that we conducted with a collaborator, Scott Hemby, who's at High Point University in North Carolina. And he trained rats to infuse self-administer morphine. And what you can see here, infusions per session and a concentration of morphine or saline, high concentrations of morphine self-administered, and once you go even higher in concentration, they self-administer less because we call it the drunk phase or the inebriated phase, where they're actually coordination and don't feel the need to ingest more drug. What we did was take the morphine vial and replace it with a mitragyna vial at various concentrations. And we could never see the animals even wanting to see there was a difference between mitragyna and saline. And so mitragyna was not substituting for morphine at many doses. And we felt that shows very low abuse potential for mitragyna itself. 7-hydroxymitragyna, on the other hand, substituted at very low doses or low concentrations compared to that of morphine, and is self-administered readily by these rats. And so we felt that 7-hydroxymitragyna has a high abuse potential and a high rewarding effect. This study was backed up by a good friend and collaborator, Jonathan Katz, who is at the National Institute on Drug Abuse itself. And he actually showed that mitragyna, when pre-dosed subcutaneously, could decrease, dose-dependently decrease heroin self-administration. And so what you can see in panel A here is heroin alone in black and self-administration up, and then it comes down just like that previous ones we saw. But if there was pre-treatment with mitragyna, what you see is a dampening or flattening of that curve. Now, if this was an antagonist to heroin, we would see the right. And if it was potentiating heroin, if it was making heroin even better, in other words, if mitragyna also rewarding and adding on to the rewarding effect of heroin, we would see this whole dose response curve shift left. We don't see that. We see a flattening of it, which is a non-surmountable blockade of heroin self-administration. From 1972 in Smith-Kline in French, but they made an observation that mitragyna by subcutaneous route was not as potent as mitragyna delivered by the oral route. I did mention that mitragyna is converted to 7-hydroxymitragyna in the intestine and in the liver. And what you can see in this is a warm water tail withdrawal test. So this is essentially, we put the mouse tail into warm water. In a couple seconds, mouse will pull that tail out of that water. Obviously, if we have something like morphine, that will allow them to not feel that warm water and keep their tail in the water. And so what we see here is a measurement of antinociception, morphine being in blue. What you can see is mitragyna is much less potent than morphine versus 7-hydroxymitragyna, which is much more potent than morphine. And pay attention to this because mitragyna is delivered subcutaneously. If we actually move forward, if I forward, and we deliver mitragyna orally, it's interesting because now you see that oral mitragyna aligns much more closely with morphine in terms of its potency. So this would lead some belief that the pharmacological activity is coming from the conversion of mitragyna into 7-hydroxymitragyna through first pass metabolism. We also wanted to look at kratom alone, just delivered orally, and what you can see is it's even less potent than mitragyna by itself, or less potent than morphine by itself as well. So we did want to make sure and confirm that this metabolism does take place. So what we did was we took rat liver microsomes and rat intestinal microsomes, and we incubated those with mitragyna. And what we looked for was depletion of mitragyna, which is the red lines. And we looked for formation of 7-hydroxymitragyna. is in black. And so the scale for grams per mil in black on the right is for 7-hydroxy, and in red on the left is for mitragyna. So you can see these are not equivalent scales, they're log scales, but mitragyna is still in much higher concentration, and we only ever see about 20 to 30, maybe 40 nanograms per mil of mitragyna and 7-hydroxy mitragyna being formed in the intestine. However, and then it's in the circulating plasma, one would assume. So we were conducting stability in plasma. I mentioned this earlier as well. And what we found is mitragyna is very stable in plasma in all species. But 7-hydroxy mitragyna was incredibly unstable in human plasma and kept converting into another compound that was the exact same mass transition. And we couldn't understand what was happening. And so to the dismay of one of my postdocs, Sham Campbell, I actually asked him to scale up our plasma reaction and add more and more 7-hydroxy to see if we could eventually isolate what the byproduct was. And the product turned out to be this mitragyna pseudoindoxyl that I talked about previously, which is even more potent as a mu opioid agonist. And we find this to be not a specific human metabolite, but an overabundant metabolite in humans compared to other species. He was excited afterwards because this ended up getting him the cover of ACS Pharmacology and Translational Science in that issue that we published this. He thanked me at the end of the day, but it was a fight to get this unknown in enough quantity so that we could structurally elucidate it. What you can see here is an insert graph above where we blew up the mouse, rat, dog, and monkey data, which is at the very bottom of this graph. So we do see this metabolite being produced in the plasma of all these species. We have yet to determine if this is enzymatically produced or if it is somehow non-enzymatically causing this conversion. Yet 7-hydroxy mitragyna to go to mitragyna and pseudoindoxyl has to go through a semi-pinnacle rearrangement, which means there should be another oxidative step in between this conversion. So we believe it's enzyme mediated. We've yet to be able to pinpoint what enzyme is responsible for that conversion. Now, the logical question would be, what are the levels that we're starting to see, right? So mitragyna being converted to 7-hydroxy and then mitragyna and pseudoindoxyl produced from that 7-hydroxy mitragyna. Are these levels really relative and are they having functional consequences within the system? And so what we decided to do a study, a PK/PD study, where we actually look at equianalgesic doses of mitragyna and 7-hydroxy mitragyna administered to rats and on hot plate analgesia tests. And we see how much of the drug was actually getting into the brain, and then how much was in the brain and how much percent maximum possible analgesic effect was present. And so what you can see in the red line here is the observed values for mitragyna administration in terms of analgesic efficacy and what the brain concentrations of mitragyna were versus the maximum possible analgesic effect. And then what you can see in this green line across the bottom is the maximum 7-hydroxy concentration that we could get in the brain. And so it was less than 1 microgram per gram of brain, which brought it down into about 5% or less maximal possible effect. And so based on this data, we drew the conclusion that the analgesic efficacy of mitragyna is not coming from the metabolite of 7-hydroxy mitragyna and must be coming from something else that's going on. And so we've been studying other metabolites that have been being generated from mitragyna, and that story is not fully understood yet, but we do know that there are more potent metabolites. We did interrogate the metabolic pathways for mitragynine, where we see CYP2D6 and CYP3A4 being the major players, CYP3A4 being the major player in mitragynine metabolism, with just minor contributions from 2D6 and 2C19. We know that the clearance of mitragynine is mediated completely by CYP3A4, and this is important because CYP3A4 is the major drug metabolizing enzyme in humans. And so there's a potential that there could be possible drug-drug interactions when someone takes Kratom and other prescription medications. We know that metabolite 2 is actually 7-hydroxymitragynine, which is mediated through CYP3A4. And this would also explain why we see conversion in the intestine, because CYP3A4 is present. It's the only CYP enzyme present in the intestinal tissue. 2D6 is also of important consequence because CYP2D6 is one of the major drug metabolizers for CNS active drugs like antidepressants, antipsychotic medications, and the like. And so this is also something that could be considered problematic in terms of drug-drug interactions. So we saw lots of metabolites from mitragynine, particularly around 31 in hepatocytes that we've identified and been working on characterizing the pharmacological activity of. So that was all mitragynine, 7-hydroxymitragynine, and mitragynine pseudoindoxyl individual compounds. But what about Kratom as a whole? I mean, this is what we're really interested in and what we really want to understand. And so what we did was we looked at the antinociceptive activity of lyophilized kratom tea. So we call it LKT. We basically brewed tea just like was done in Malaysia with leaf material. And we freeze dry that, and then we resuspend it and deliver it in a uniform measure to these animals. And what you can see here is morphine, either by the IP or PO route, being almost equivalent and producing 100 analgesia at the same doses. And as we move from 45 milligrams per kilogram to 4 grams per kilogram of lyophilized kratom tea, we never really exceed 60% analgesia. So there's a ceiling effect that seems to be associated, maybe some self-regulation with all those compounds that are present within Kratom, not just looking at mitragynine or metabolites of mitragynine. We also see with Kratom tea at the various doses, we can see analgesia lasting almost as long and comparable at the highest doses to that of morphine. And so, you know, it's no surprise that this could be used as a substitute for pain control when we compare kratom tea preparation to that of morphine. We wanted to really understand what receptors were responsible for the analgesia of kratom tea. And what we learned through utilizing opioid receptor knockout mice is that it seems to be a combination of mu opioid and kappa opioid. And I won't go into detail for the sake of time. I realize we're already quite over time, so I will try to get to the end here and wrap up. We do know that lyophilized kratom tea lacks a conditioned place preference, and so if we want, and anybody has a question afterwards, I can go into this in detail. But essentially, what this tells us is that Kratom does not have a drug-seeking behavior in animals. We also know that Kratom does not seem to cause sedation in animal models. And that's just showing you data here. Interestingly enough, morphine doesn't cause sedation in low doses either. But a U5488, which is our control, is a kappa opioid agonist known to cause sedation. But lyophilized kratom tea doesn't appear to cause any sedation from baseline activity. And then one of the major we wanted to look at was respiratory depression. because opioids are known to cause respiratory depression. See respiratory depression with morphine, but in the case of wild type mice, we see a little bit of respiratory depression in the first 20 minutes with 1 gram per kilogram, but that comes back very quickly, and we don't really see that issue in the more knockout mice, or even in the more knockout mice treated with a kappa antagonist, although you do see a little bit of respiratory depression prolonged in that state. Overall, we don't think that the respiratory depression is nearly as much as you would see with opioids, and we don't see issues with sedation. We also looked at this in withdrawal assays, and so we habituated animals to morphine over a number of days, and then we precipitate withdrawal with naloxone, the opioid antagonist. And if you look at morphine here by itself, one of the measures that we look at is jumping frequency, and these animals are jumping. We call it actually popcorn jumping because they literally look like popcorn just popping off of the floor. In the different studies where we looked at 4 days of morphine sulfate or 7 days of morphine sulfate, and then 4 days of kratom tea, or just 7 days of morphine sulfate and 1 dose of kratom tea, 4 days of morphine and 4 days of kratom tea in decreasing doses, or 7 days of just kratom tea by itself, you can see that we see very little opioid withdrawal signs in those animal groups. One of the other things we can measure was teeth chattering. Just like when you're cold and your teeth are chattering, these animals will do the same thing. We see a very robust teeth chattering with morphine, now precipitated withdrawal, and you can see in the different groups that was decreased when compared to just morphine by itself. So I mentioned that mitragyna is the major alkaloid, and indeed, in most of the products that are available in the United States, vendors are looking for mitragyna to be the major alkaloid in the product that they would put into the market. So this is what we see, and this is a product that we purchased in the United States. We made a tea from it. It was dried leaf material, but what you can see here in this lyophilized tea by percentage weight, mitragyna is the largest concentration. Speciosa was the second most abundant alkaloid, followed by speciogynin. And interestingly enough, 7-hydroxymitragynin, as I mentioned earlier, is below the limits of quantification. And so we don't see 7-hydroxy even in many of the products now that are being sold in the United States. That could be because the product is turning over faster. It's being sold. It's not sitting around. It's not being stored for a long period of time. It's really moving quickly through the marketplace here now. So this is pharmacokinetics, or exposure to the different kratom alkaloids that we see after dosing this lyophilized kratom tea. And we can see mitragyna is the major. Speciosa is the next, which is another isomer of mitragyna. 7-hydroxymitragynin is present in red. You can see it there. It's the third most abundant. And that's interesting to note because it's coming completely from metabolism of mitragyna. And then we see corinanthidine, which is another compound that's very low in concentration, but it appears to be higher in bioavailability. And so what are these compounds? The second most abundant that we see in exposure, and this follows up by in humans, and I'll touch on that in just a moment. We do have some human data now. Speciosa also binds to mu and kappa opioid receptors with much higher affinity than does mitragyna. And I'll remind you that the only difference between Speciosa and mitragyna is the position of this hydrogen. And so in mitragyna, it's going into the screen with hash marks. So it's away from you, what we call the alpha configuration. And this way it's coming out towards you in the beta configuration. And what that does to the molecule overall is it completely changes the shape. So mitragyna, all of these 4 rings are basically like a flat piece of paper. But when you switch this substitution of this hydrogen into this, where you see Speciosa, it actually bends that whole molecule into a more L-shaped configuration. This is important because that L-shaped configuration seems to fit more efficiently into the mu-opioid receptor. And so Speciosa itself seems to be a very good mu-opioid agonist as well. And again, it's the second most abundant compound. We also see compounds called penanthine and speciogynin. And I'll just jump to speciogynin. The data is almost identical. The only difference between speciogynin and penanthine is the reduction of this double bond. Speciogynin is another mitragynin analog where now this exocyclic ethyl group is in the alpha configuration instead of the beta configuration that we see. What's fascinating about this is it converts the activity from being opioid and adrenergic to now serotonergic and mildly opioidergic. Very high affinity serotonin compounds at 5-HT1A and 5-HT2B. 5-HT1A receptors are important because they've been shown that stimulation of those receptors will actually combat opioid-induced depression. We talked about this being a beta arrestin pathway mediated phenomena where we're seeing this sort of specific signaling at the mu-opioid receptor, reducing the side effects. But at the same time, it may be the fact that in Kratom as a whole, we have activation of these 5-HT1A receptors, which also are working against opioid-induced respiratory depression. Then we also see serotonin 5-HT2B receptors, and those 2B receptors are known as anti-drug targets. They're involved in cardiovalvulopathy. If you activate them, luckily what we've learned is these are antagonists at 5-HT2B receptors, and so there is no cardiovascular concern, at least from activation of that receptor subtype. So penanthine and speciogynin both have about the same activities, and interestingly enough, really become important in terms of their abundance at the outset because they're additive. We also wanted to see if any of that serotonergic activity resulted in analgesic activity. And just to make a long story short, penanthine and speciogynin both cause antinociceptive activity that can be reversed by 5-HT1A antagonist way 100635. So these are contributing overall to the analgesic effect of Kratom as well. So I showed you already opioid, adrenergic, and now serotonergic activity involved in the analgesic effects of Kratom overall. The first human clinical pharmacokinetic study was published earlier this year in March by friends of ours at Washington State University. And what they did was gave a single 2-gram dose T to some patients, and they saw some of the exposures in the blood supply for those patients. And we're working with them actually on looking at a lot of the metabolites that have been exposed. But this really opened the door up in a huge way in the United States for studying Kratom in humans. This was the first one in the US that got published. There are a couple of other trials that are underway in North America. One is a FDA trial that has been awarded to a company called Alta Sciences. That trial is reported to be on track to begin in early 2023. They're going to be looking at a safety and tolerability with single doses. And then they plan to do a human abuse potential study with established safety, which in some ways is kind of ironic because the FDA has been trying to remove Kratom from the marketplace because they say it's unsafe. But here they're funding a trial to establish its safety and ability. But it is what FDA needs to do. They need to figure out where the safe levels of this product are and what can be utilized in a safe manner in humans. There is a Kratom plant study that's been done in Canada, and to my knowledge, this has ended. But this was probably the largest controlled human clinical trial with Kratom that we know of at this point. And we're anxious to see the data come out. Some of the data was presented just in June this year at a meeting of the College on Problems of Drug Dependence. And all they showed was the objective opioid withdrawal scale. So a subjective opioid withdrawal scale and the clinically opioid withdrawal scale. And what they showed was absolutely no withdrawal. It's interesting. These were patients that only took for a certain, so it's not really a comparative study to someone who's been taking Kratom for a long period of time and if they would experience withdrawal. So I'll wrap up, and I'm sorry I've gone long, but I really wanted to go into a little more detail than I usually do, particularly around things that are happening in the United States versus what we see in Southeast Asia. So we know there's been a long history of safe use in Southeast Asia, but in the West, we've seen a wide range of adverse events. I hope that you believe me that the alkaloids are very structurally different from opioids, have different pharmacokinetic and pharmacodynamic products. And I hope that I convinced you that the products in the US are not the same as what we see in the traditionally used, what I call fresh leaf preparations in Southeast Asia. And that's something I think that really moving forward with products that are coming into the US is going to be important to really pay attention to. What are the chemical differences between fresh products and dried products? And therefore, we still have gaps around science, and we have a lot more work to do to fully understand what's happening. But one of the things that is certainly encouraging is that a possible solution to the opioid epidemic could come from nature, where opium came from originally. And so with that, I will end. I'd be happy to answer any questions. And thank you for your attention.
Well, thank you so much, Professor Chris, for your time and also your talk. It's really nice to know about this one. So maybe the other team can help me to give the share screen for the question and answer, because we accepted also a lot of questions actually. Okay, wait a moment.
Sure, and I would love to hear. I don't know how I don't at all how Kratom is being used in Indonesia. So it would be interesting to hear.
Okay. Mm-hmm. Okay, so the first question is, could you please tell us more about Kratom's medication-assisted therapy, and what kind of product is evaluated that result in the less than 9 swallows that you mentioned earlier in your presentation?
Yeah, so this product that was evaluated was the product was a dried leaf material in a capsulated form. So it was the true leaf material. It wasn't a concentrated extract. It wasn't some manipulated product. It was just the leaf material that had been dried. That's pretty much what I know about that study. We weren't involved with that study. And a little bit more about the medication-assisted therapy. I don't know what more you want to know, but we feel, at least something that we've believed in for a long time, is this is where we're hoping to move a Kratom product into studies in humans to see if we can really utilize it as a way to move patients off of clinically used opioids and as an alternative to something like methadone or buprenorphine, and using it in that treatment setting.
Okay, so thank you for the answer. So maybe we can move on to the next question. So there are three questions. So the first one is like, any reports about a microorganism relating to kratom alkaloid synthesis or anabolism in a kratom plant?
It's a fascinating question. It's a great question. We don't know of any reports. That doesn't mean that it doesn't exist. We have been working with two different natural product biosynthetic chemists. One is Sarah O'Connor, who's at the Max Planck Institute in Germany, and the other one is here at the University of Florida. And we've been looking at gene clusters specifically in the biosynthesis pathways, and they've almost figured out the entire biosynthetic pathways and enzymes related to that. But that doesn't mean that there's not microorganisms interfering with that process, and that's a fascinating question, and we don't know the answer to that yet.
Okay.
With the second part of the question, let's see.
Yep.
How about
Mitragyna or derivative residue concentrated in the blood and liver within time?
So we know that it's eliminated at a fairly constant rate over time. So we don't see it concentrating, and we haven't published it yet, but we've finally done some multiple dose pharmacokinetic studies where we can get to steady state with Mitragyna, and then we give a last dose of Mitragyna, and then we follow that for several hours, days afterwards in terms of blood supplies. We don't see accumulation of the product. The product is pretty well constantly eliminated after it's at steady state. And so we haven't really seen concentration, and we don't see accumulation of the metabolites either. So I hope that answers that question.
Okay, and maybe the last part of the question is like, is Mitragyna linked at the same site of opioid receptor as morphine and cocaine?
Yeah, so that's another interesting question. We know that Mitragyna is binding we believe in the same site of the opioid receptor as morphine, but it's not as morphine bind, and so we don't see the change in the receptor conformation as drastic as what we do with morphine, and that could be why Mitragyna does not activate the receptor as potently as morphine does. And cocaine, I don't know. I mean, I know cocaine doesn't bind to the opioid receptor, but one thing we don't know is Mitragyna bind to the dopamine transporter or any of the transporters like the serotonin transporter, which cocaine is known to bind primarily at serotonin, norepinephrine, and dopamine transporters. So we don't know how Mitragyna could be interfering with or decreasing cocaine or methamphetamine intake at this point.
Okay, thank you so much for the answer. And the next question is like, you mentioned before that kratom is mild or lighter than opium and morphine in terms of addiction. But are there any other side effects, of consuming it?
Yeah, well, it's interesting because I want to underscore the fact that what we have seen are very acute dose situations, right? So we've looked at these in a number of days in animals, not over months, not over years. And what we have seen, at least in the United States, they've started to come out with a kratom use disorder diagnosis for humans. So we are seeing some individuals that are becoming addicted to kratom over time, and they're having a very difficult time getting off of kratom at the end of the day. Now, it's also difficult to really understand what that is because we haven't seen this in a clinical setting. So I'm not sure if these individuals are taking kratom in a traditional sense or if they're taking it in these concentrated extracts, and they're building up a tolerance and taking more and more and more and more, and so that they get into a point of physical addiction that we would see with a traditional type opioid or stimulant. But is that really kratom? So what we call kratom in the United States is anything from that leaf material all the way to these very concentrated extracts. And so it's still a little bit confusing in understanding what is causing the adverse effects, because we haven't been able to study these under any controlled clinical settings in detail. And so I can tell you, I have meetings with physicians that treat addictions. They are treating opioid addiction with kratom in the United States. It's not approved by the United States Food and Drug Administration, but it's being used to treat individuals in the United States with opioid disorders. At the same time, those same physicians are treating individuals that have trouble getting off of kratom as well. So there seems to be this sort of paradox and this sort of double-edged sword into really understanding where the safety is, where the efficacy is, and then where the abuse or the misuse potential comes into play. So yes, there are some adverse effects consuming it over time.
Okay. And maybe for the last question, is there any difference? Yeah. Is there any difference in the chemical composition and effects for kratom leaves grown in the US compared to the one that from Southeast Asia?
Yeah, this is a great question. And in the beginning, we really felt that there was a tremendous difference because we were seeing, as I mentioned before, we were seeing trees that were producing very low levels of mitragyna in the trees that we were having in the United States. But we started to also find some trees in the United States that were donated to us from people's individual collections that then were producing high amounts or very similar profiles to what we saw in marketed products. And then when we went to Malaysia and we actually sampled trees within the same farm, we saw that diversity was there. And so we're really not seeing a tremendous change in alkaloid composition of what we're growing in the United States now versus what we believed earlier. We published a paper, I think in 2009 or somewhere around there, about the different alkaloid profiles in the United States grown kratom versus traditional Southeast Asian grown kratom. And I think that paper we sort of outdated ourselves on now, and we've learned more over time, and certainly it is fascinating. There's several differences in the United States. I alluded to a little bit with the soil. We don't have the richness in our soils we have in Southeast Asia. We don't have intensity of sunlight that you have. The growing season is not the same, and so there are various drawbacks to trying to produce kratom in the United States that would not be at the equivalent of what you would see coming from Thailand, Malaysia, Indonesia, in areas of the world. And so these are some of the other consistencies that we're trying to see what's happening.
Oh, I'm sorry, Professor. I think it's getting late in your case, but we still have 4 more questions. So is that okay?
No problem.
Okay, so another question is like, is there any restriction on mitragyny research due to the drug content, which is considered illegal in some countries?
Yeah, I'm not sure about other countries. I mean, we've been collaborating with groups in Malaysia and in Thailand, which of course it was more recently legalized in Thailand again. And in Malaysia, it's still criminalized, and they haven't decriminalized it in Malaysia. Yet they're still allowing research to be conducted. So I don't know the details of the laws there. In the United States, we almost faced a situation where to be made in 2016, but luckily the government decided that they would invest money in research instead of closing the door and not allowing research to go on. And so we've been fortunate to be able to do that. And we haven't had any restriction in terms of moving research forward here.
Okay, so it's actually another one is we have a 4 papers on Indonesian kratom strong enough to be funded by BRIN, hopefully. So he asked if it's possible to have a collaboration with you on the Indonesian pharmacogenomics team, something like that. So maybe later on we can share your email probably, Chris, so that he can directly contact you and then have a talk with you.
Yeah, absolutely.
Okay.
As I mentioned at the beginning of my talk, it would be really wonderful to have some collaboration. We don't have any other than the products that we assume are coming from Indonesia that are in the marketplace in the United States. We've never had access to fresh material or known source plant material from Indonesia, and so it would be great to do genetics, to look at how these compare to those from Malaysia and Thailand, and really understand if there are differences, if there are similarities, what all is happening. So yeah, we would welcome those opportunities.
Okay, and another question is like, is there any compound that is similar to mitragyny in terms of pharmacodynamics action, or is it completely unique, not identical to cannabinoids, morphine, or cocaine? Well, it's
I think that the most exciting thing for me is that it is, as far as I know, is completely unique. The pharmacology is just fascinating, and from the very first day that I saw our panels come back where we tested it over all these central nervous system drug targets, I said, oh my goodness, it's interacting with everything that we use to treat drug addiction. And so this, to me, in one molecule was just a really unique thing. The only other molecule that I would say comes close to this is ibogaine, and ibogaine is from the iboga species in West Africa. And it's a completely different species and plant material, but it also, interestingly, has been studied for substance use disorders as well. Okay. But totally unique compared to cannabinoids and opioids or stimulants. Yeah.
Okay. So maybe for the last question is like, what are the benefits and function of kratom alkaloids for the human body? I don't know, maybe he meant like the whole alkaloid.
Yeah,
so
no,
it needs to be
this is really, it's been a fun fun project for us because we've actually been able to isolate enough of the other alkaloids aside from mitragyna and understand some of the pharmacology associated with those. You start to look at molecules activating serotonin system and adrenergic system, and all of these systems can help with mood elevation. They help with pain. So there's some potential benefits on many of those. There's also been some work done to show benefits to the nervous system overall with health. Also, benefits for regulating blood sugar and blood pressure. So there's many anecdotal reports that still don't have much science behind them to really understand what's happening. But we also have to be careful that we don't think of Kratom as a panacea or a cure-all. Many people seem to tout it as being so beneficial in so many ways, but we really need to let science guide us and tell us where the benefits are. So, and where the dangers are. It's not all beneficial.
Yeah, that's true. Okay, thank you so much, Professor Chris, for your time and also your talk and discussion. So I think I will firstly ask first to the audience if they want to ask directly. And if no one raised their hands, so maybe we can. Oh, is there any? Okay, then maybe no one. Okay, thank you so much for your time. It's really wonderful session and also all the talk and discussion. So it's really nice to have you here to give a talk.
Yes, thank you all. And I'll put my email in the, well, maybe. Well, you can share my email with everyone. And if anyone has specific question or specific collaborative ideas, then please reach out.
Okay, thank you so much. So yeah, that's all for today's webinar. Thank you so much for all the participants for your timing and also the time for the discussion. So I will also close the webinar session for today. Thank you so much. And maybe enjoy and wait for another webinar from our research center group. Thank you so much.
Thank you so much. Bye-bye.
Bye.
Mbak Ani, silakan dilanjut.
Ya. Oh, bahasa Indonesia ya, Dek?
Boleh, silakan.
Baik, terima kasih semuanya yang sudah hadir untuk webinar kali ini. Mungkin seperti yang dikatakan oleh Profesor Chris tadi, kalau misalnya ada pertanyaan lanjutan, bisa dikirimkan secara langsung ke email yang mungkin akan kita share ya di kolom chat untuk semuanya, sehingga kalau misalnya ya sudah di-share, mungkin kalau misalnya ada pertanyaan tambahan atau ide kolaborasi, dipersilahkan untuk menghubungi beliau langsung. Mungkin demikian webinar kali ini. Saya mohon maaf kalau misalnya ada kekurangan dari kami tim webinar, dan juga terima kasih atas partisipasinya. Selamat siang untuk semuanya. Terima kasih.
Terima kasih.
Terima kasih Mbak Ani, terima kasih semua partisipan sudah ikut hadir. Izin live ya.
Terima kasih Bu, izin live.
Izin live.
Ya.
Terima kasih Bapak Ibu semua, izin live.
Terima kasih, izin live.